Thank you for joining my presentation of my lab's work at Grand Valley State University, evaluating the impact of microbial experience on immunity. The immune system is a dynamic network of different cell types and that ultimately all work together to protect against infection. This is composed of innate immune cells like the neutrophil and the monocyte and macrophage, as well as adaptive cell lymphocytes like the B cell and the T cell. My lab is interested on a range of these immune cells. We know that various environmental factors impact immune function and development. My lab is interested in these environmental factors. We know that diet, family dynamics, environment of being that the child grows up in, as well as other factors impact the immune system. My lab in particular is interested in microbial experience, which basically means which microbes and how many of these microbes is the immune system exposed to, and how that impacts immune development. Interestingly, we know that environmental factors also experienced by the astronaut during spaceflight impact immunity. We know that gravity changes, radiation, and stress can induce a broad immunosuppression, so decreased immune function in an astronaut during spaceflight. Interestingly, we also know that an astronaut is exposed to pathogens during spaceflight due to confined space. When we look a little bit further at this, we also can see, based on previous reports, that certain viruses show increased reactivation and increased viral shedding in spaceflight, and that certain bacteria and fungi show increased rates of proliferation in space than they would normally on Earth. Ultimately, what this leads us to hypothesize is that an astronaut experiences an altered microbial experience. What we don't know is how that altered microbial experience impacts immunity, especially in terms of a long-term impact. My lab uses a unique mouse model to study how microbial experience impacts the immune system. We use mice that are both specific pathogen-free, which means that they have very low microbial experience, as well as mice that we purchase from pet stores, which have much higher microbial experience. We co-house some of the specific pathogen-free, or SPS mice, with pet store mice. During this time, pathogens from the pet store are transferred to the SPS mice, therefore altering their microbial experience. We analyze these mice at various time points during cohousing, and we also do extensive pathogen screening for viruses, bacteria, and various parasites, and more detailed immune profiling to see how these changes in microbial experience impact immunity. Here's some of our preliminary data. What we see is that over time, our co-housed mice in the red gain experienced T cells over time, whereas our specific pathogen-free mice do not gain these T cells over time, indicating that our model is working and that we have altered microbial experience in our co-housed mice, and then this results in a change in the immune response. For our future directions, as we continue to work on this, we are very interested in measuring systemic inflammation over time, determining innate immune changes, as well as adaptive immune changes over time, as well as directly testing immune function.